In this lecture, I'm going to talk about immunity to protozoan infection. And as you know that protozoans are unicellular eukaryotic organisms. They cause diseases in human, including amoebiasis, Chagas disease, African sleeping sickness, malaria, leishmaniasis, toxoplasmosis. And immunity to protozoan infection, it depends on the location or site of multiplication of the parasites. For example, intracellular parasites, they are subjected to cell-mediated immune reaction, and the extracellular parasites, I mean free parasites, or within the bloodstreams, they will become target for antibody immune responses. And I will highlight some examples of immunity to protozoan infection. I will start with giving a short account on immunity to leishmania, which causes leishmaniasis. And leishmania lives in phagosome of macrophage, so it is intracellular parasites, and all intracellular parasites, they are target for cellular immune responses. So immunity to leishmania is mainly T helper 1 cell, as a T helper 1 produce interferon gamma that kill intracellular A mastigotes. And now I will just also highlight possible mechanisms for parasite neutralization of Plasmodium falciparum asexual stages. So these are different ways that can kill malaria parasites. For example, antibodies or antibody-mediated phagocytosis, as can be seen here. These are antibodies. They bind to infected red blood cell with Plasmodium falciparum, and as can be seen here also monocytes, they have receptors known as Fc gamma R1 and Fc gamma R3 receptors. These receptors are specific for IgG1 and IgG3. So when the infected cell captured by IgG1 and IgG3, this will bind to the antibodies. This will bind to the receptors to Fc gamma R1 and Fc gamma R3, and this actually lead to antibody-mediated phagocytosis. And also phagocytosis could be complement-mediated, as can be seen here. Also monocytes, they have some receptors for the complement. So when the red blood cell infected by Plasmodium falciparum parasite coated with complement component, they come in contact, actually they bind these receptors, and this will lead to complement-mediated phagocytosis of the infected red blood cell with Plasmodium falciparum parasites. And as can be seen here, these are macrophages. They produce some chemical intermediates, including nitric oxide, tumor necrosis factor alpha, and these they have parasiticidal effect on the intraerythrocytic parasite or myrizoid. In addition to that, antibody also has the capacity to interfere with the development of intracellular or intraerythrocytic parasite. And so, as can be seen here, these antibodies, they bind to some antigens on the surface of the infected red blood cell. So this will lead to inhibition of intra-erythrocytic parasite development by these antibodies. However, these antibodies also, they have, as can be seen here, they have the capacity to capture or to bind the parasite through the leaky schizont, as a schizont or schizont. Prior to rupture of the red blood cell, it becomes leaky, and within these cracks, these antibodies will enter and neutralize or capture the parasites. Also, antibodies, as can be seen here, they have the capacity to enter to parasitophorus bed. So this is a red blood cell, and this is a parasitophorus bed, and these are parasites. They are now neutralized or captured by this antibody, and this actually known as antibody mediated inhibition of intra-erythrocytic parasite development by antibodies entering through the parasitophorus duct. But however, following rupture of this late stage of infected red blood cell or schizont, the free merozoite also, they will become target for inhibition by antibodies, and so these antibodies, they interfere with infection of or interfere with infection of red blood cells by these merozoites. So now I will highlight African sleeping sickness, a disease caused by trypansoma species. But however, the most important is that we are going to highlight trypansoma brucei, as this trypansoma, they contain around 1,000 variant service antigen or glycoproteins. So this will lead to escape of effective immune responses by what is so-called antigenic variation, and this help in removal of the majority of these parasites. But however, around 1% of parasites escape killing by following a process of antigenic variation that enables the parasite to escape immunological clearance. And as can be seen here, these are the nerve fibers. So trypansoma multiply in the blood and processing to neurological stage and infect the central nervous system. This will cause to meningoencephalitis and lead to the loss of consciousness in this disease. And also one of the protozoan disorder caused by trypansoma cruzi, it is known as Chagas disease. And actually our concern here in this lecture is to show immunity to protozoans infection. So in case of trypansoma cruzi that has the capacity to cause Chagas disease, the immunity start as an innate immune response, and this is actually very important, as the dendritic cells, they produce interleukin number 12, and this will shape the adaptive immune responses. So the immune response will become T helper 1 and CD8 disease and CD8. But however, cytokines by T helper 1, for example, and CD8, they produce interferon gamma and tumor necrosis alpha. This may lead to cardiovascular lesion and mental disease in Chagas disease. So now I am going to talk about toxoplasmosis, and this is caused by a protozoan parasite known as Toxoplasma gondii. So immunity to Toxoplasma gondii starts by vigorous innate immune responses, and these innate immune responses at the beginning of the infection is very important, as innate immune cells, they produce some cytokines that shape the adaptive immune responses. Notably, dendritic cells, they produce interleukin number 12. And innate immunity, so it is the first line of defense against the infection by Toxoplasma gondii, and by producing interleukin number 12 from dendritic cell that shapes the adaptive immune response together with T helper 1 in controlling infection in the early stage by interferon gamma. So the infection is controlled by interferon gamma at the early stage, but at the late stage, the infection is controlled by the CD8 cells or T cytotoxic interferon gamma. However, toll-like receptors, they take part in immunity to Toxoplasma gondii, and this actually takes place early in the infection. And as you know, that toll-like receptors, they are known as pattern recognition receptors. They recognize pattern associated molecular or pathogen associated molecular patterns, and maybe toll-like receptor number 11 are 12 implicated or shown to be effective in toxoplasmosis. Thank you very much, Professor Ahmed.